All right, this is the podcast for cell theory and microscopes. Make sure you have your podcast handy, and we're going to get started. So let's talk about the history of cell theory. Now, for centuries, scientists had no idea that the human body was made of trillions of cells. And it wasn't until 1665 until a man by the name of Robert Hooke made a simple microscope and looked at a piece of cork. Now, he called what he saw cells because it reminded him of the cells which monks live in at a monastery. Now, a cell is the basic unit of all living organisms. During the late 1600s, Anton van Leeuwenhoek designed his own microscope. He saw living organisms in pond water, milk, and various other substances. Seeing living organisms in milk probably wasn't too good, knowing that there's living things that you're drinking, consuming milk. Today we have to worry about that because it's been pasteurized. But anyway, continuing. In 1838, Matthias Schleiden carefully studied plant tissues and said they were made of cells. Theodor Schwann found animal tissues also consist of individual cells, and Rudolf Virchow in 1855 proposed that cells come from existing cells. So we have a lot of names here and a lot of parts of cell theory, but that's why we're breaking it down, and we'll be going over this in much more detail in class. Now, all these guys contributed, all these scientists contributed, and this led to the cell theory of today. And there are three parts of cell theory, and it states these three things. The first thing it states is that cells are the smallest living thing. If it's smaller than a cell, then it's not considered living. Part two, all cells come from pre-existing cells. And the third part of cell theory says all living things are composed of one or more cells. Now, remember, a theory is not a guess. A theory explains why something is true. So cell theory explains the explanation of cells, and it's these three things right here. A theory is not a guess. That would be a hypothesis. A theory explains a law, and a law is simply something you observe. Okay, so microscopes is a tool that all these scientists use to help develop cell theory. Now, developments in microscope technology have given scientists the ability to study cells in greater detail than early scientists ever thought possible. A compound light microscope is one of them, and it uses a lens and light to magnify an object. That's why it's called a compound light microscope. It uses light. Now, scientists often stain cells when using a light microscope because cells are so tiny, they're thin and translucent. By staining them, it's much easier to see the detail inside. Now, the problem with compound light microscope is that objects scatter light and that blurs the image. This is a disadvantage of a light microscope. The maximum magnification of a light microscope is around 1,000 times magnification. And when we're working with things that are really, really small, sometimes we need greater magnification than just 1,000 times. So there are some non-optical microscopes which do not use a lens, and they needed greater magnification to study the cell even deeper. Ernst Ruska developed the electron microscope in the 1930s. Now, it aims a beam of electrons at thin slices of cells. This is known as a transmission electron microscope. It's going to form a black and white image, and the reason it does that is because electrons do not come in color. So it's going to form a black and white image. Now, it can magnify up to 500,000 times, but the specimen must be cut very thin. And because you're cutting it very thin, it's going to be dead. Now, a scanning electron microscope, not the transmission, but a scanning electron microscope directs electrons over the surface of the specimen, providing a 3D image. Now, with the scanning electron microscope, only non-living cells and tissues can be observed, so this is a drawback. But there's a newer type of electron microscope called a scanning tunneling microscope. The scientist holds up a probe to a specimen, and electrons come out of this tunnel from the probe. That's why it's called a tunneling microscope. It can get 3D images from things such as small as atoms. So we're talking super, super small here. And it can also be used with live specimens, so it has some advantages over a scanning electron microscope as well as a transmission electron microscope. Cells. Now, cells exist in various shapes and sizes. We all know what cells are. We know they make up our body. They make up living organisms, but they have some things in common and some are different, but they have some things in common with one another, which we're going to look at now. Now, all cells, regardless of what type they are, have one thing in common. They all have a cell membrane. They also have DNA. The cell membrane isn't just the one thing they have in common. They have some other things too, but they all have a cell membrane. And we call this, it's also known as a plasma membrane. So if you're hearing the word plasma membrane, that's basically the same thing as a cell membrane. Now, this cell membrane controls what enters and what leaves the cell. Most cells have genetic material in some form that provides instructions for making substances that the cell needs. Cells also break down molecules to generate energy, because when you're not generating energy, you die. No ATP means death. All right, there are two basic cell types. It's not plants and animals. A lot of students like to categorize them as either a plant or an animal. Those aren't the two basic cell types. They are prokaryotes and eukaryotes. So prokaryotic and eukaryotic are the basic cell types. So let's break down these words and talk about what they actually mean. So the U in eukaryotic means true. Pro means before, like prototype, for example. That's before the product's release. You have a working demo. And you have kary, which means nucleus. So when you combine these words together, it's very easy to figure out what they mean. Eukaryotic means it has a true nucleus. Prokaryotic means it doesn't, because the cell existed before the nucleus evolved. So eukaryotes literally means true nucleus. These cells have a nucleus. Prokaryote literally means before the nucleus. These cells have no nucleus. Eukaryotes are 100 times larger than prokaryotes, and because they're so much larger, more complex, they have many internal parts known as organelles. Now, organelles are specialized membrane-bound structures that carry out specific cell functions at different parts of the cell at the same time. This would be the nucleus, the ribosomes, the lysosomes, the endoplasmic reticulum, whether it be the smooth or rough ER, lots of different things that we'll be talking about in section 4. So we're going to learn about all the organelles and what they do. All right, so here we have a prokaryote. Here we have a eukaryote. And in this eukaryote here, we can see there's lots of detailed smaller parts. This is what we call the organelles. Over here, we only see a few small detailed parts, but there is no central nucleus here. They both have a cell membrane or a plasma membrane that controls what enters and what leaves the cell. But this is a prokaryote over here. This is a eukaryote. This has no nucleus, while this one does. This one is also much more complex. They both do have DNA though. So this prokaryotic cell over here, this is a eukaryotic cell over here. I know this is an animal cell because it is circular and less square. We'll talk about that in class. All right, moving on. So eukaryote and prokaryotes. Now, eukaryotic cells are defined as cells with a nucleus and other organelles that are bound by membranes. We just talked about that. The nucleus is a central organelle that contains the cell's genetic material in the form of DNA. Now, multicellular organisms made up of eukaryotic cells are called eukaryotes. You are a eukaryote. All right, your cells have a nucleus. And because all your cells have a nucleus, you are a eukaryote. Now, some unicellular organisms like yeast are also eukaryotes. A lot of times students think they're prokaryotes. No, you are a eukaryote. Now, prokaryotic cells are smaller and more simple than eukaryotes. These have no nucleus and no membrane-bound organelles. And most, it says most, not all, most prokaryotic cells are bacteria. Now, many scientists think that prokaryotes were the first organisms on Earth. According to the endosymbiotic theory, a symbiotic relationship involved one prokaryote living inside the other, with both benefiting. Eukaryotic cells have organelles that have developed specific functions, which has led to cell diversity and thus more diversity in eukaryotic organisms. Without eukaryotic cells, life would not have evolved to be more complex than bacteria. So that's it for the intro to cell theory and microscopes. If you have any questions, please leave a comment below, or please bring your questions to class. We're going to be doing something with this. So make sure you have all your notes, and we'll see you guys in class.